Again, a very good evening and a warm welcome to the fourth lecture, which is on the risk assessment. And here has been the outline for the lecture for this session. Overall, like this session has been like we do have two sessions been coming from the risk assessment. The initial one, which covers the basics onto the introduction and the history, and then covering some couple of the major sections, which has been talking about the hazard identification and the dose response assessment, and followed by the other two steps, and the recent updates in the risk assessment field will be captured from the second chapter. So here is my question: like, what does actually being a risk assessment mean? Is it been really been pertain only to safety or the toxicology, or is it been a general sentence? It's been everywhere, right from you take about your finances. Any of the companies got the finance risk management and or the risk assessment, and similarly, like whatever the industry we take, we do have it. But when it comes to the toxicology, like we do call it as a toxicological risk assessment. And let's see like what was actually being the introduction in the history context of it. So it all started in 1958 with basically the Denali clause, which actually banned the carcinogens in food. And later to that, like many of the analytical advances led to the tolerance and development of the tolerance and the acceptable risk levels. Sometime back in 1970, the US Congress basically created an science-based environmental laws, like which was actually been the foundation for risk assessment. And moreover, in 1983, there has been actually been an NRC, which is actually being gone by the US government. And basically, NRC released a book which actually defined the framework for the risk assessment, which includes the major four steps. What we see over on the screen, they are the hazard identification, the dose response assessment, the exposure analysis, and finally the risk characterization. And basically, in simple words, like people even call it as an red book. And okay, so here you see basically the development of the NRC assessment, like the guidelines which have been scheduled provided over the years. It was started back from 1983, and basically the most, I think, if I'm up to date, then basically the most up to date information might be from 2017. I heard something in 2021 and 22. There has been some update been going on. Not sure whether that was being clearly completely published. So these are the different books which have been actually been published for the risk assessment paradigm. And however, two of them are really very important, and they are literally been called not by the versions or not by the year, but by the color of the book. It sounds funny, but that's how it is. The red book, why we call it, has been because it's been in red in color, and similarly the silver book, the 2009, it literally also shaped the feature of the risk assessment from 2009. So the red book and the silver book have been the important ones, which actually laid the foundation for the framework for the risk assessment. And moving on, basically, like there are a total of when we talk about the risk assessment frameworks, they were actually been evolving over the years. As we talked about, saying like something as a foundation was packed back in 1983, and then basically it's been 40 plus years now, and literally like the things have been evolving. And basically, for example, the past focus was literally being onto the cancerous risk, and in some cases, like in most of the cases, not some, like it would be the single chemical single pathway approach, like what happens if this chemical interacts with this person or the environment or the specific population at this particular situation. And similarly, like the stakeholder input was been mostly from the government back. And later towards to the modern approach, this actually been expanded to the horizon towards to the non-cancerous one, and which actually includes the lifetime calculations and also the ecosystem risk, and basically which integrates the latest omics, like where what we call from the last over the decade or 2 decades, like all the omics, like proteomics, metabolomics, or even including the biomarkers from the mol bio and so on. And the modern approach also includes the multi-pathway and multi-endpoint analysis. And most important, unlike the past one, like basically where actually the government is the one who is to identify the chemical risk and then basically publish a document, there was actually been a forum being created by by like a sort of a task force, or the forum being created, so that the government can actually work along with the corporates and hand in hand, where the companies come in to actually maintain a stakeholder engagement and try to complete or address the gaps for the toxicity, and try to come up with the final reports. When it comes to the definitions, basically, here has been the simple pictogram what actually shows like what's been an hazard and the risk. And basically, like if you see on the left hand side, let me turn on my cursor pointer. Just give me a second. Okay. If you see on the left hand side, on the like the man just been watching the ocean from the beach. Basically, he's actually been trying to, like, what he sees is been there's a shark in the ocean, and that's been an hazard. So something that can actually potentially cause an harm can be an hazard. But back, like when it comes to the risk assessment, we actually talked about like what exactly has been the risk. So basically, like the probability of the exposure to that hazard is being a risk, basically, which will cause the harm. So towards the right, what we see is been saying like there has been a guy who has been actually been, maybe we can call him as an dare guy, and basically he has actually been trying to swim along with a shark in an ocean. So what we see over here in this picture is being this is the hazard. What we see like where we do have the shark in the ocean, and basically like when it comes to the exposure, like this is what we try to explain. Like risk always is being equal to hazard times exposure. When someone exposes to that hazard, that's where basically we try to get back into calculation of the risk. And similarly, like on the next slide, what we see is been over the saying like this can be a beautiful pic. This has actually been a very old pic. This was actually just from the textbook. So maybe the image might not be clear. Like I can just explain you like what happens is being like there were two couples actually being on to a beach in the morning, and they were actually enjoying the sunny day in the morning. And however, towards to the afternoon, here has been the clock like which shows like it's been the afternoon, and one of them actually got an umbrella which actually gave them some shade, and they've really been still enjoying the beach atmosphere. But the other one is actually being charred because there was no control over there. So in simple words, like what we try to see the risk assessment is being sort of an umbrella which actually protects the people from that particular hazard without having a sort of an issue. And moving on, here are some more examples. Uh, Moving on, here have been a couple more examples when it comes to the hazard and the risk, like basically like water being the like water and like maybe the gasoline, they have been the hazards. But someone basically being without knowing the swimming, just jumping into the water can be actually be a risk. And similarly, and so on with gasoline and the matchstick. These have been the analogies to being used just to make the things much more easy and set the stage for the early toxicologist before we jump in. And this has been an interesting slide, and I just tried to because we have been in the technology of AI and the ML, and basically I just tried to give my story to the ChatGPT and said like, okay, can you just create me a sort of an image? What image can you create out of it? It was not so good. It literally used my text what it is with just a glass of water. But I can just continue with the storyline. Like what really happens is being like basically like there's been a guy who actually went for a walk in the morning or maybe a jog in the morning, sunny morning, and he really had a good time during the exercise. Like it's in the morning, and basically it's a summer day as we said, and he was literally been sweating and dehydrated. And once he is back to home, like he literally quenched a glass of water and he's done with it. But basically that's good, which actually is being good. But what happens is like coming back to the point of dose makes poison from our previous lectures, where the authors actually or the speakers have actually clearly communicated like why dose makes the poison, and which is actually been by the as said by the Paracelsus. So someone actually who's been drinking over the liters and liters of water over a short span of time can literally even create like it can be toxic. So why again? It's basically imbalances the delicate salt balance and so on, and it has the water intoxication as well. So in simple words, what is not toxic? Like even water has been toxic. So that's the whole story behind the story of water. So coming back and jumping into the details of the risk assessment. I've actually tried to pick up some of the snips as the flowcharts as such from the different contexts, like the guidelines. The specific one is being from the WHO guidelines. Basically, what we see is being the context is not more important for us, but basically what the main point I would like to explain over here is being about like when we talk about the human risk assessment, it all started from the environmental risk assessment. So basically, the environmental risk assessor is the first framework which came into the picture, on which basically the scientists in the industry developed the human risk assessment. So this is the flow like which we would be seeing in the coming slides as well. Basically, I just want to show like this is what exactly is being the birthplace for the human risk assessment. And moving on, here has been the framework, what we will be talking about, saying we do have we have actually talked about four major steps, which are being the framework for the risk assessment, which are being the hazard identification, dose response assessment, exposure assessment, and the risk characterization. Apart from that, the first main step which actually comes into the picture is being the problem formulation. So basically, whenever we talk about an issue, what we see in industry has been something different to what actually the government or maybe the industry people a few decades back they actually tried to solve the situation. For example, we can take a talk about the arsenic, or we can talk about the dioxins, or we can talk about the fluorine. So there have been many examples there, but basically, be- before jumping into the risk assessment, what is been really needed is being the we need to have a clear formulation of the situation, like what has been the objective, and basically like where we are with the approach, and what exactly should be the scope of the assessment, and basically like finally the risk management goal, and finally moving on to the chemical identity. So once we are done with the formulation of problem formulation, then basically we jump into the hazard identification. So basically like when we talk about the hazard identification, we try to see like what exactly have been the potential hazards of this chemical to a human, or maybe even to the environment, and so on. And then from there we try to see like the hazard characterization, or maybe basically we try to see like what are being the are there any existing guidelines which actually already been derived some values there, and basically if yes, then what were being the assumptions or the scenarios that basically were being considered for the specific scenario. Then followed by we try to jump onto the exposure assessment. Over here what actually we try to say or show is been about like having the assumptions basically how much it is being exposed, how long it's been exposed, or who are actually been there exposed, whether it's been an infant or a geriatric or a pediatric or an adult or adolescent or a pregnant woman, and so on. And then finally coming back to the risk characterization. Here basically we try to estimate the exposure and finally compare with the actual endpoints, what were being the guidelines, or maybe what can be the RFD, or maybe the slope factor, or what, or the POD, or the NOEL. It can be of any of the parameters. That's how it moves on. And this is actually again one more important framework for the risk characterization. Basically, like again, this is actually been for the reference for the people to just see like how this flows. And the again coming back to the members like who are actually been wondering, we talked about the framework and we directly jump into the frame, like the toolkits, like we're actually, or the flow charts. Not to worry, the things have been coming front, and over here like what I'm trying to showcase has been the background approaches, what really made a move for the human risk assessments. And again, coming back, why a risk assessment is being needed. this can be an $1 million question, saying like what has been the real need for the risk assessment. um, because you can see like basically we are the humans who have been in the center of the pyramid, and we are actually being surrounded by all different types of exposure scenarios, and where it can be a common food and beverages. What happens is being fire through ingestion we actually get exposed, and again from the soil and water, and again from the ingestion and also from the dermal we get an exposure, and from the air we again actually have the exposure from the inhalation in the dermal, and to the last the consumer products, where actually we do have all three types of inhalation like exposures like ingestion, inhalation, and dermal. And this is one more schematic diagram which actually shows a big picture and says like how nitty-gritty we can get into the details. Like for example, if when we talk about the soil or the things, basically the exposures can be from the plants and basically the fruits and vegetables, and then again from the ingestion and so on. It can be from the animals, or it can from the meat and the dairy as well, or the groundwater, or even right from the indoor air. So again, these have been the exposure factors that which actually comes into the contact, and these have been the exposure routes. So setting that context, here has been the risk assessment paradigm in practice. So what really happens when it comes to the risk assessment process? The first step is being the systemic review. Systematic review, when we talk about this is been where actually we try to see the problem formulation, and we try to actually go with all the details, including the existing research, including the epidemiology, including the clinical studies, and also the in silico models. So once we are actually having that's been the input for the risk assessment, then basically what we see to the next step is being the hazard identification. So once actually the hazard been identified for that specific chemical, okay, this has been actually having a carcinogenic risk, then basically we try to come back to the next step called dose response assessment. Over here, what we try to calculate is been saying like till now we actually got the qualitative data saying like okay, we do have the risk with this specific chemical, but how to get the quantitative data, like basically at what level it causes the risk, or basically like what's being the concern at which situation or which species of the animal, or maybe like what not, like there can be multiple if and buts there, but basically like when it comes to the quantitative assessment, that's where we talk about the dose response assessment. Then again, the output from the dose response assessment would be the basically the parameters what we try to call like the NOEL, LOEL. It can be the RFDs or RFCs or the slope factors for the cancerous chemicals. So this is what actually been the input for the toxicity values, and then basically we try to move on to the next step called the exposure assessment. Till here, what we actually seen is been saying like, okay, there is a chemical which is actually having a concern. This has been the hazard, and now the hazard has been characterized. Okay, this has been the safe value, or maybe this has been the POD for this chemical. Then basically we try to come back and see like, okay, if there has been a device, like if there has been a product which actually been exposed to that specific person, then basically like how much that has been exposed and how long that has been exposed, and basically talking about like what are all the different populations that comes into the exposure. And finally we actually try to derive the estimated dose concentrations. Then finally coming back to the risk characterization, this again drives to be the quantitative data. And finally we try to see like how many times the exposure is been, whether it's been onto the higher side or the lower side, what we call in just simple words the margin of exposure or the margin of the safety. So this is what actually we saw in the risk assessment process. But however, on the other side, we do have the risk management process. So this includes like what the outcome from the risk assessment would be the input for the risk management decisions. So basically, maybe the toxicologist who has been driving or authoring the risk assessments on the bench would really be driving the these many departments of the specific corporate or the maybe the scientific body of the government to take care on the next steps of the actions. It can be political or it can be legal. If there's been any concern, like the concerned ingredient or maybe the chemical, that can be legal as well. Right now, we do hear a lot about PFAS, or maybe we do hear a lot about BAPs and so on. There can be all-time favorite ingredients which are actually been falling in this batter. And at the same time, we do have like the outcome from the risk management can also provide us the control options. It doesn't mean or it doesn't end saying like, okay, this is what has been happened, and if this has been the situation, that is how actually one need to dwell going forward. No, that's wrong. If there has been a concern, then basically we can tighten the values of that specific thing or control it by either with the mechanical or the actual chemical values, and then basically we can try to filter it. That's how the Caliprop 65 works with the drinking water. And so from here, we'll be moving forward a bit deep into the different steps, like what are being the different four steps that we talked about. I hope like till now, like what has been covered have been the different frameworks and the structures. And from here, we would be moving a bit more into the details about the each actual steps. Coming back, what we're trying to explain is been like till now, like we actually try to cover the different frameworks. And from going forward from here, like we'll be dwelling more deeper into the each individual part of the section of the details, starting with the hazard identification. So basically, like when we talk about the hazard identification, it's basically in simple words, basically we call this to be an assessing the detailed toxicity of the chemicals. And then basically like when we talk about the toxicology paradigm, like what was actually being discussed, if you remember from the yesterday's lecture of Pradeep, basically where we actually saw the different mechanisms, and for that specific mechanisms we actually need to have the different touch points. What we see or what we say has been like starting from delivery, which actually includes the absorption, distribution, metabolism, and excretion, and to that of the interaction, and then moving on to the cellular dysfunction, and then basically the biomarkers, and then the disrepair. So basically, like when it comes to the hazard identification, there are four different subsections of the hazard identification, and which starts from the SAR, the structure activity relationships, and then basically we talk about the in vitro and the short term studies, and then we move on to the in vivo animal bioassays, and then finally we conclude the hazard identification with the human epidemiological studies. So this is the paradigm, like what we actually saw from today's presentation. This has been the framework that what we are trying to express when it comes to the hazard identification. Like this should be the framework, like how actually the mechanism of action that works in order to that to be a toxic. So moving on, basically like coming back to the hazard identification, like why the SAR, the structural activity relationship, has been an important. As we all know, like being in the industry, and we actually come across with different chemicals and on a day-to-day basis. And there are so many chemicals, like where actually we don't have the complete full set of available data. And there were actually the regulatory efforts to how can bridge that gap. So, for example, if there has been an situation in order to characterize the risk, if the studies are being done, that's a different scenario. But just in order to identify the hazard, again completing the full set of studies, which could really incur billions and billions of dollars, would not make sense. So from that per se, in 1998, basically EPA has actually come up with a list called high production volume chemicals. And out of them, basically the 43% of the chemicals were lacking the public toxic data. So as a response for this, basically like they actually got an tie up with the industry. That's where we talked about like the past versus the future, where actually we say like we do now have the equal and very good hand by the industry as well, while framing or while navigating the situations, especially with regards to the chemical safety. So over here, there were around 2,200 chemicals being tested, and obviously, yes, I said like these were being industry sponsored, and EPA, HPV, along with tie up with hands with OECD SITs. That's again an international efforts where actually the OECD was harmonizing the guidelines. Could actually come up with along with the EPA to move this much faster as well. So moving on to the one more additional or the big block that actually which could actually build this one was being the REACH framework. It was initiated or drafted back in 2003 and finally approved it in the Parliament in 2006, and from 2007 that was being implemented. And that's where basically what we try to see is being like as a part of the REACH framework, like one need to actually submit the chemical safety dossiers to the government in order to place any of the chemical into the European market. One need to submit those dossiers to the European government, which should include the right from the physical properties to that of the human toxicological data endpoints and also the ecological toxicological endpoints. And over here, the focus was actually being the non-animal tests, especially while encouraging the possibility of moving towards to the in vitro and go moving away from the in vivo, and lately also developed as an in silico. And at that point of the time, like the GHS labeling was being very nascent, and later to the maybe almost like a decade later that came into the effort. But that's where it started with the GHS labeling. And also, there has been one more initiative from the US where actually we do have the unit called the TASCA, and they actually try to create an integrated chemical environment database aiming the 21st century for the toxicological updates, which moving towards to the in vitro from the in vivo. These were the backgrounds, and basically like coming back, basically like why actually the SAR is being need to identified at the early stage. If you do have the different 4 or 5 chemicals, and then basically like if while coming with the different set of chemicals, we need what we need to see is been about the structure or the physical properties like the solubility, stability, and basically the electrophilicity and the volatility, and also the chemical reactivity. So these are the factors which actually could drive grouping the chemicals, and we can see a couple of the good examples over here, like the which are actually being known as a renowned carcinogens or the potential carcinogens. Maybe we can call it as per the textbook language. So which are being the aromatic amines, like they are being the highly regulated as an occupational carcinogens. And similarly, like wherever we do have the amino acidides or phenanthrene nuclei or the nitroso and nitroso groups, so basically those are being the structural alerts, which gives us a pop-up saying like, okay, if there is no data, then basically maybe we need to have some additional details because they can be a carcinogenic risk. And similarly, like QSR has been growing largely into the other sections which are actually been into the nanomaterials and also the EDs, which are being favorably called EDs, the endocrine disruptors. But however, there's been a challenge being remained. Basically, like the read across under the reach is being actually the what exactly they try to move is being saying like group the different like the similar chemicals together, and then basically provide a complete collective risk assessment, and maybe they're going to do go with the collective assessment. Coming back, here are being the details like what we see is being about this has been again a strip from the textbook, and these are being the requirements for any of the pesticides to be placed in the market, like what all the different types of studies and what all the test systems that need to be done, and basically like what would be the cost incurred. So if you go back and see like what exactly have been the costing for the CARC studies have been literally been the $2 million. So similarly, like like especially with the developmental neurotoxicity, it has been literally been costing like $700,000. So overall, this would literally cost from up to somewhere like I mean, billions of dollars moving from just to perform the risk assessment. So moving from here, what exactly are being the connector circles of the boxes with the help of the complex mixtures? We can literally be group them together, all the related compounds, and including the nanomaterials and the EDs. And then basically we can actually come up with a sort of toxicity equivalent factors, which are actually being for the especially for the aryl hydrocarbon moieties, where the AH receptor mechanism is being known. And that's where the WHO actually pushes towards to the international TEF values as well. And we do also see the success and challenges. The success over here is being though the SAR was actually been an initial tool that actually been tried to make use of the grouping the chemicals, but we had a very great success with the pharmaceutical industry, where actually they actually had and one more layer on top of the SAR for the ligand designing and the ligand binding, where the 3D modeling and along with the combinational chemistry, which actually could help them to identify, fit the receptor of interest, could identify the right molecule, and that's where the success from the pharma industry came in. And however, unfortunately, or maybe the current situation, or maybe the present situation is being of, especially for the environmental risk. We do have very limited data about the receptor binding success, so that's been one of the challenge. And moving forward, again, there has been a push from the reach to see okay, what about the nanomaterial screening, or basically how can we repurpose this situation for the EDs, because EDs have been well known for receptor binding assays. So we can talk about the estrogenic receptor or maybe the androgenic receptor and so on. So basically we try to repurpose back again what was been the success from the pharma was actually back pushed for the even for the chemicals, especially for these niche segments. So, moving forward, as said, what in simple words, what we try to see is been about saying we identify the similarities, and then basically we look for the common features, the common functional groups or the precursors or the breakdown products, and then basically we group them as a category. This has been at a very superficial level of just to give a big picture, saying how we can make use of the SAR. So, moving from here, we talk about a couple more steps onto the in vitro and the short-term studies. So, basically, in this regard, we talked about the Tox 21 approach. So, the 21st century toxicology paradigm was being basically they were trying to push for the short-term test assays, which can actually have an impact on the risk assessments. For example, we can talk about the evidence of a non-mutagenicity from both the in vitro and the in vivo short-term assays. Basically, can have play essential role there, and where actually the regulators can consider a non-linear carcinogenic risk assessment paradigms for non-carcinogenic carcinogens. I'm sorry there. So non-genotoxic carcinogens, what I mean. So basically, in this specific situation, what all been the essays that were being included? It's basically it's the AIMS essay, where we see the reverse mutations, and then basically we do have identify the forward mutations with the help of the mammalian cell mutation assays, which also includes the in vivo cytogenetics. And basically this was actually been extended to such an extent where we can actually come up with developmental repro, neuro, and even the immunotoxic assays. So one of the one of the big phase, or maybe the one of the big challenge over has been about development. And on the other hand, the biggest other than maybe rather than being the biggest one over the validation, over the development is being the validation. So for validation, we do have the two entities came forward from US, and one from US and one from Europe. They have been shortly called ICC Aquam and Aquam, and they support the 3Rs, which actually stand for reduce, refine, and replace animal use. So over here, they tried to develop 40 plus validated alternative methods over the years, especially starting from the different endpoints, driving towards starting from the low risk, that is the cosmetic segment, and also towards to the ending, starting with the localized assays, especially with the corrosivity or maybe the sensitization for the and including the toxicokinetic parameters. So, especially over here in this case, there are many international efforts being made by the global countries as well, including the Korea, Canada, Japan, along with these two entities in order to reduce the number of animals for the chemical safety testing. So, as an outcome for that, these are the guy like basically these are the in vitro tests which actually came into the picture. So, what we see are being for the irritation. These were actually being developed and also being validated by the OECD, and this is which includes the episkin and basically the and basically like we do have the MATEC epiocular and so on, the BCOP, and even the corrosion test for the Corsitax. Moving from here, we will jump into the animal bioassays overview. So, the which actually plays a very key role within the hazard identification. So over here, the EPA and the IARC actually developed the guidelines where actually they say like just like our Indian penal codes, unless you have been proven guilty, basically like that means like we need to capture it, that's find them as a guilty. So similarly, like our the EPA guidelines, what they say is being like unless it's been proven the otherwise non-carcinogenic, then maybe we need to consider them as a toxic. So that's where the whole thing comes from. And basically, the IARC has actually worked around with 900 plus agents, and out of which like 400 chemicals were being classified into the different sections of probable, possible, carcinogen, and basically like or carcinogenic. We will see like what does that probable, possible, and actual carcinogens mean in the next slide. But that's how the basically they have been classified, and the basic principle remains the same. Like if we see animal tumors in animals, there can be a high chance of replacing the same thing in the humans as well. So that's where the basic principle comes from, and that's again a precautionary principle applied by the EPA. Moving on to the specific things like the cancer bioassay design. Over here, basically like it was being insisted or it was being recommended to maintain minimum 2 species and that too with both the sex of the animals and minimum 50 animals per dose group and that too near, especially for the cancer bioassay, like we need to have the near lifetime exposure. And more importantly, the strains, number of doses and the dose levels are also being important. And again, the dosing would be typically ranging from the 10% to that of the 90% of the MTD, max tolerated doses. And finally, the endpoints, major endpoints could be the tumor count or the, and especially it's not only the tumor count, it's sometimes it would be interesting to see like what's the latency, like how the latency has been reduced to develop the tumor or what exactly have been the rate tumors or maybe the total number of tumors across the body. And even the NTP advanced to the next stage of going even an inclusion of the pre and post, that's the perinatal and the postnatal, which includes the gestation and lactation as well. So over here, what we see as an limitations for the cancer bioassays, firstly, high dose issues. Basically, like only highest doses show tumors, and almost many of the occasions it was the near dose that which caused the systemic toxicity as well. So, but however, on the other hand, like there were some compounds like were actually being non-toxic, but at the same time they were found to be saturated the metabolism, or maybe the saturating the elimination pathways at a high dose. So with this, they actually came to and they have recommended rodent concordance like where actually it could be 70% between the rat and mouse. And that's where at particular section they have been recommended to go with the male rats and the female mice as well. And interestingly, we do have a couple of the exclusions to the thumb rule of the paradigm saying like if it is been and carcinogenic in animal, this could turn carcinogen into humans as well. So let's see those examples in the next slide. And one of the example over here is being about the aflatoxin and where actually there has been identified of 1 lakh times difference between the sensitive rats and the resistant mice strains. And also, as we talk about the mechanistic differences, it can be even based on the SIPs or maybe the pathways or even the enzymes which actually could take role into it. And beyond cancer, we do also have the mechanistic integration. So what exactly is being needed is being the combining the data. So we do have the short-term tests, and we do have the data from the biomarkers, and we do have the epidemiology. So basically, what we need to do is been something like this. So here has been the example like aflatoxin, we even induce liver tumors and DNA edicts. So basically, like wherever we actually see both of them, the DNA adducts along with the liver tumors from the aflatoxin, there has been a clear relationship where the relationship is being turned to be the linear in case. And also like this enables the low dose extrapolation. And for the non-predictive rodent tumors, there were different cases. Like these were actually the examples that we are trying to explain. Not all fits into the thumb rule. Here have been the exclusions, especially with the what was actually being the carcinogen in male rats were not within the female rats, vice versa, and the same thing has been extrapolated to the humans. Because what we see here over here is being like here is the table. Like you can see the renal tumors in male rats, and especially induced by the unleaded gasoline or maybe the 1,4-DCP, shortly called like DCP, which is the dichlorobenzene or the D-limonene. And where they try to bind with the alpha 2 globulin, a specific minute protein present within the male rats and not been identified within the female rats. So this specific issue has been identified within the male rats, but found not the same with the female rats or any of the species, including the human. So similarly, we talk about the saccharin. Saccharin has been, and again, a best example for the rodent exposure, where actually we see the hypoplasia being identified. And the same thing was not been true again, coming back to the humans. Why? This is where what we see the two different cases, that is the qualitative cases and the quantitative cases. And similarly, we do have the example for the titanium dioxide, and we do have many more examples which we can really read from the table. But a couple of them are being called out into the text, like ethyl acrylate and so on. So these have been the examples or the which actually could be the outliers which break the thumb rules, like something being carcinogen in animals doesn't need to be a turn a carcinogen in humans. So this is the one of the important way how actually we can eliminate that situation and go with the better value or the maybe the select the right POD while doing a risk assessment. So moving forward, we do have the emerging models and the genetic tools as well, where we talk about the transgenic mice, and basically we do have the a specific section called collaborative cross mice, which actually been a fully sequenced models been available, and also the human cell models. These are actually being provided. But as you see on the screen, like there's been a note, the transgenic mice models were actually being primarily being used for the mechanistic characterization rather than the hazard identification, because that involves a lot of cost as well as the time to the industry. Moving on to the last step of the hazard identification, which is being the epidemiological data in the risk assessment. So why actually we do go with the epidemiological studies is being which actually could provide the strong evidence for the human risk assessment with when we actually the data is being found. There were three types of studies where we do see the cross-sectional studies, the cohort studies, or the case control studies. So cross-sectional studies, what we see is being it's not the causation study, where that means like we don't get to see like what's the cause and what the effect. It's basically the survey groups, and we identify the associations and see like what's been the outcome of it. And with the next one, the cohort one, like what we see is being the exposed versus the unexposed groups over the time, like the treated group versus the control group. And similarly, the case control, like we actually compare the exposure history of the deceased versus the non-deceased groups. So over here, there are a couple of challenges again. So basically, like the primary challenge is being the retrospective exposure estimates, which was actually been a big challenge. And one other thing is being like the to get to the see what exactly been an issue within the human, we need to actually capture the long latency times, because carcinogen like something as a compound might not turn into a carcinogen over in days or weeks or months. If it has been, then that has been in a very high potent one. But however, usually compounds would take some months or years of time to get to see or send some signals where sort of an toxic effect being seen. And similarly, like at the same time, the multiple chemical exposures were being identified over the lifetime. And then finally, the confounding lifestyle factors, especially which includes the smoking and diet and even the alcohol as well, consumption of alcohol. So now moving from here, like when we try to evaluate the epidemiological evidence in the risk assessment, we need to consider a couple of the findings over here, like what's been the strength or what is the consistency of the specific report over the time and again, and what's been the specificity. And similarly, like what exactly has been the biological plausibility and so on. And basically, we do have even some good tools in place, which is actually the meta-analysis, which includes the combines the results from across the studies and weighed by sample size. So we can see some of the examples over here, especially from the arsenic or the dioxin risk assessments from the EPA. And moving on to the last next step, or like which way where we could try to call like the most recent advances in the field of epidemiology. So nowadays, like basically, like what we see is being like emergence of new trends, just like what we see like the booming of AI in the recent times. There was a time like where actually there was different niche segments of science, like the molecular biology or genomics and the proteomics, the omics group and the epidemiology came together to form the molecular epidemiology. So over here, what we see is being the integration of different things. And then basically, what actually made the advances or maybe what were being the tools that was been driving it. So some of the good examples were being the single nucleotide polymorphisms, which have been shortly called like SNPs. They were being identified, and basically the genomic profiling were being done, and basically the transcriptomics and the proteomics were also being a part of it. And especially with regards to the FDA, they try to track the drug response gene data as well. So the benefits of from this, basically what we see is being the improved biological plausibility and better cross species comparisons, especially when compares to the animal to human data, like the rodent to human data, and then basically systems based understanding beyond linear models. So over here, we try to integrate the qualitative aspects into the risk assessment. And over here, like what we try to see is being like qualitative assessment generally includes the consistency of the species or the experiments or the endpoints or the adequacy of the experiments to detect an adverse effects and so on. And when it comes to the classification of the systems, like we talk about, this is where I said like some time back, we would be seeing like what is the possible, probable, and carcinogenic compounds. So here is the classification from the EPA and the IARC. These are the entities which actually provided into the four classifications, where we do have the sufficient data to call it as a carcinogen, or where we do say like it's limited data to call it as a carcinogen, or inadequate data to call it as a carcinogen, or no evidence to call it as a carcinogen. And basically, at the same time, the EPA similarly calls them as a likely, suggestive, or inadequate, or not likely. And the application of these sections would really be more falling into the, it's not only the CARC, but also the repro and developmental assessments as well. And moving on to the mechanism of action and the AOP adverse outcome pathways in the risk assessments. And back in 2005, within the EPA guidelines, like weight of evidence came into the picture, and they tried to put this forward to identify the key events and process. So, for example, consider the pesticide mixtures. If a group of pesticides within the different insecticidal properties all inhibit the acetylcholinesterase enzyme, their cumulative risk can be assessed by summing up their individual HQs. But at the same time, if they are there are endocrine descriptors, consider call them like the chemicals with similar effects on the estrogen receptor, which can be grouped for a cumulative risk assessment, and again considering them for their combined impact on the reproductive health. So that's where the situation, or maybe the explanation from the MOA and the AOP came into the picture. And basically, the MOA framework again considers the different steps, like it comes from the animal studies, and that should be the human relevant, and obviously that should be the lifetime assessed, especially for the carcinogenic chemicals. And when it comes to the uses, like the chemical specific adjustment factors, interspecies differences or the human variability, and the uncertainty factors would literally been called out over here. And then basically again for the there's been a good criteria by Hill, which they call for the causality, like the dose response and the temporal associations and the biological plausibility and the coherence and the consistency and the specificity will need to be for basics for the MOA. When it comes to the AOP, the adverse outcome pathways, it was introduced back in 2010 by Ankli and et al. And again, I would like to emphasize the point saying like this was again for the ecological risk. And so basically like that means that was extended to the human risk at the later stage. So even the OECD was being completely inspired by this development, and then they actually pushed a lot towards building the AOPs and framework linking the biological events to the adverse outcomes. And that's where they try to even develop so many things, especially with the space of sensitization. So this is the picture, like this is the pictogram what we see over here, like for the especially for the AOPs. This literally goes again as a sort of a subsection of what exactly the molecular mechanisms lecture that what we saw yesterday, like right from the delivery to that of the disrepair. So what we see is been over here is being like the toxicant, and basically like where actually it is been exposed, and then basically like what are all been the key initiating events, and then basically like what are being the biological responses, and then finally what's the disease. So this has been good for the mechanistic approach, but what's in there for a risk assessor? That's what we need to see over here. So if you see like the assessment tools or the methods, so especially with the exposure and the key initiating events, like basically the chemical properties, we could actually retrieve the details from the QSAR. We could identify that from the QSAR in silico tools. And similarly, the toxicity pathways, especially for the biological response, we do have different in vitro and the in vivo assays over here. And then finally coming to the disease patterns, then where actually we see the regulatory endpoints. Those have been the adverse outcomes. So finally, moving from the hazard identification to that of the second stage, which is the dose response assessment overview. So within the dose response overview, like basically, this has been again one of the important and quantitative aspect of the risk assessment. Like basically what we try to see over here is being like the basic dose response assessment, and then the exposure assessment, then the susceptibility variation and uncertainty characterization. So over here, traditionally in the past, like even the US EPA used to actually come with a thumb rule saying like anything non-cancer endpoints can be a threshold-based approach, and basically like anything that's been a known carcinogenic endpoint, like then that can be a non-threshold based one. But the emerging trends actually proved that maybe yes or not to a set of 100% wrong, but however, there has been a possibility for the threshold approaches for some non-genotoxic carcinogens that need to be identified. And why actually there has been a extrapolation challenges, like we do have very limited human data, and obviously like we try to rely mostly on the animal bioassays, especially for example with the environmental chemicals or the day-to-day chemicals that what we see. And one of the other extrapolation where we see is being like the doses, like where actually we're trying moving from high dose to the low dose, or maybe the animal data to the human risk, and so on. So the focus over here is being to get below the observable experimental responses. And that's where the critical effect identification comes into the picture. So firstly, select the robust data with the lowest observed adverse effects, and define the critical effect, basically, which could really call that significant adverse biological effect at the lowest exposure. And for how to do that, that basically like we do have the very good guideline from the WHO, which is actually called the principles for modeling dose response for risk assessment of chemicals. So this could be further referred for the detailed step-by-step approach. And over here we talk about the threshold approach. Over here we do try to see like what exactly have been the POD. So to the right hand side, like we do see a graph over here where we do see on the dose versus the percent response graph. And we do see a curve over here and where the points like F, G, H, I are being the doses that are being tested. And it was identified basically the F to be the Noel, which was actually being the highest dose without statistically significant value, and G to be the lowest dose with the significant value, which is again that can be called as low. So the POD was derived at the ED10 value. So that's where we see the small square box. That's the E value. And what's the significance of it? So basically that depends on the number of dose levels tested and number of the animals tested in each dose, and also includes the background incidence for the adverse response versus the control group. So over here, that's what we try to see. And we also try to see the different curves, A, B, C, D. What do they mean to us? So basically, like the curves A to D show some options for extrapolating the dose response relationship below the range of biologically observed data points and the POD. So that's what it mean. And moving from here, like what exactly should be in the factors that have been calculated for the non-carcinogenic risk. We can consider the examples for reference doses or the acceptable daily intakes. And here is been the formula for the RFDs or the ADI. The formula is being the same, like Noel divided by uncertainty factors or times modifying factors. And please note, there's been usually there's been a like assumption saying like ADIs and TDIs are same. So ADIs are being the acceptable daily intake, and basically like TDIs are being the tolerable daily intake. They do have some minor issues, but they are not acceptable. So that there's been a difference between that ADI and TDI. And what exactly have been our uncertainty factors? We would be seeing in the next slide what are the different factors that we can consider for. So we do have the interspecies factor, intraspecies factor, and then basically like moving from the different dose responses, like what was been our POD. If it has been a NOEL, that's a factor 1, and basically if it has been a LOEL, then basically we do have the additional factor of 10. And similarly, like for subchronic to chronic or subacute to chronic, and from the short-term studies to the chronic studies, we do have a different factor. And at the same time, we do have some modifying factors as well. Modifying factors can be adjust actually the uncertainty factors based on the availability of the mechanistic or the kinetic data. For example, consider like there has been a similar metabolism for a chemical which has been identified in both rats and humans. There is data which could actually say like, okay, this has been the chemical exposed to both the rat and humans, and they have the same pathway of metabolism, and also the final endpoint would which has been the same, which would be the same active target metabolite. Then basically we could like address, or maybe we could actually adjust the uncertainty factor 10 for the interspecies to that of the 3. There can be a question saying like why 10 to 3. So this is where we see this picture. The uncertainty factors develop from here, like for any of the like species, like within the animals, like basically like intraspecies, the species differences would be, we do use a factor of 10. And again, the 10 is being for say calling out for including both the toxicokinetics and the dynamics. And similarly for the human variability, like intraspecies, again we use a factor of 10. So that's again, it's the over here it's the 4 for kinetics and 2.4 for the dynamics, and over here it's 3.2 times 3.2. It's both the same for kinetics and dynamics in humans. So in addition, we do see a different population groups over here, right from an infant to that of an geriatric. So basically, like especially for the pesticides, the Food Quality Protection Act actually tried to add an additional factor of 10 when actually they are trying to assess it the same risk assessment for the any of those sensitive populations, including the pregnant women. So what we see onto the next step is being the MOE, like the margin of exposure, as we talked about, saying like it's nothing but the novel divided by the human exposure. So here there's been an example. We can just take an example of saying like we can come back to the person who has actually been jogging in our first slide and looking out for a glass of water. So assume like that person actually has been drinking a water which contains a compound at a neurotoxic compound which is actually been available so like where the NOEL was identified during the dose response assessment as 100 mg per kg per day. At the same time, the total daily intake of the compound, maybe he might be drinking consider like 3 liters or 4 liters, and assume like this has been the amount that has been actually been going in per kg per day. Then basically what we try to do is been the human exposure. The MOE is been nothing by NOEL divided by human exposure. That's the value of what we see is been of 2500. So in general words, like what we actually try to see on the back slide over here, the 10-10 factor, we have not adjusted the heat over here. So in simple words, like basically what we try to say is being like, okay, MOE greater than 10 reflects the 10 times 10, which includes the interspecies and intraspecies. So that's where if something be less than 100, then that would trigger the regulatory attention. So, saying that Noel has been a widely used approach over the decades or from the inception for the risk assessment, but they do have some limitations as well for the Noel approach. Basically, like why and when it came into the picture, when actually the alternative risk assessment, like the tool, like the PMD came into the proposal back in 1980 by the EPA. And basically why again basically like Noel would be the one of those test doses, and basically like when it comes to the Noel the complete dose response curve might not be completed if the Noel has been identified, unlike the benchmark dose what we'd be seeing in the next slides, like how that would be populated, and basically how the POD would be derived as a BMDL 10 or 5 we would be seeing in the tools. And over here, the tools to be identified, like EPA actually got a BMD software, which again is been for both the carcinogen and the non-carcinogens. And the advantages over here is being the ability to take basically the account for the dose response curve, and basically again it includes the measurement of variability, and it actually come up with a consistent BMR like benchmark response that has been calculated for the RFDs. So in this approach, the dose response is modeled and the lower confidence bound for a dose at a specific response level. The BMR is basically being calculated, and the BMRs can be specified at different rates, like it can be BMD1, like a BMR 1, 5 or 10%. And this specific graph talks about the BMD 10 and the BMDL 10. BMDL 10 stands for lower confidence limit, and basically which is actually been a part of the ED10. Moving forward, like we do have two main approaches for the within the specific test. Basically what we do have is been the statistical model, and we do also have the mechanistical model. And when it comes to the statistical model, like we assume each individual has actually a threshold or the tolerance for any of the known probable chemical, or maybe the distribution, where it can be normal distribution, like where it can be a probit distribution where the curve turns to be S, and where it can be a log logistic model which actually turns to be similar to probit. Here the calculations or would be something would make a mathematic genius over here. I have not just captured the equations over here. And similarly, the mechanistic models, basically, which would literally build the equations based on the biological mechanisms. So what does that mean to a risk assessor? Basically, it's just not the statistical fit, but however, it's basically based on the biological mechanisms. It can be identified into the one stage hit or one critical event leads to an effect, or multimodal, or even the multimolar linear stage as well. So, which is being called like LMS model by the EPA, which where they try to use the this particular specific tool for the quantitative cancer risk assessment, especially for the inorganic arsenic from the soil, where they try to use the linear multi-stage model. So this has been an interesting snip again from the CND. So basically, like this here has been an example of comparison of the ingestion of arsenic from the soil. But however, the difference is been between the A and B is been like the A talks about the point estimation method, where the numbers are being fixed, and basically like, okay, this is the soil concentration, this is the ingestion rate, this is the exposure duration, and this is the exposure frequency. And here are the body weights for the specific population, and this has been the averaging time for the exposure. Then finally times the bioavailability, then times the final the availability of the specific compound, that's the Q factor. So over here, like basically when we talk about the other way around, why actually being the Q is been important. So the Q is being the slope factor for the lifetime risk over here. So that's where the final outcome calculation would give us the lifetime cancer risk for from the skin cancer for the arsenic from the point estimation method. But if the same thing, same compound, same exposure, it's been actually been captured from the probabilistic methods. Here are the numbers have been completely different, because these are there are tools which actually try to go bring up some accurate numbers, or maybe the titled numbers here. So over here, what we see is been about again the 95th percentile exposure. The 95th exposure is being like somewhere like 2.3 times 10 to the power of minus 5 versus the final like the lifetime risk, like what we see about the 1.4 to the 10 to the power of minus 4. So here are some of the commonly used terms, like, or maybe the abbreviations for the non-cancerous and the carcinogenic endpoints, like what can be the TDI, or what can be the PTWI and ADI and slope factor and BMD. I just try to capture this sniff again from the WHO document, which could make the ease for the readers at the later point of time while preparing for the ABT exam. And that's it. So basically, like from here, what we have covered till now has been like we try to cover like what exactly has been the introduction and the history, and what have been the definitions in simple words, like what was been the hazard and what was been the risk, and what's basically the decision making paradigms. We have seen different flow charts, like what are actually been trying to, like how actually a risk assessor work on those things. Then finally dwelling deep into the two of the major steps for the hazard identification and the dose response assessment. And the following two sessions, like basically like the exposure assessment and the risk characterization and the emerging concepts, and with some of the good couple of examples and real-time examples would be delivered in the upcoming lecture. Here are some references, and before I close, maybe before I say, like I would like to share snips of these two textbooks. Someone who's actually been an beginner, I would recommend to start with this. Like basically, this would be a very simple textbook where actually you could get the essence of the risk assessments. And then basically for pros, obviously, yes, CND has been the Bible, as we all know. So that's it. Thank you.